But let's get started anyway, because I don't want to drift too late. I'm very happy to introduce Pieter Roelfsema, he's coming to us directly from Amsterdam, and he's going to be speaking on control of synaptic plasticity in deep cortical networks. Guess we want to put Pieter up on this. Oh, does he manage the combination of pictures? Okay.
Okay, can you hear me?
Yes.
Okay, great. So I'm very sorry that I can't join you. What happened is that I applied for the European visa waiver program because I have a new passport, and then I had to fill out a couple of questions. One of them whether I had been in Iran. I actually was in Iran last December. And they don't give you feedback about the success of your application. So last Sunday when I was checking in for my flight, I couldn't check in. And the reason I found out later is that my visa waiver has been rejected. I suspect it's because I was on this meeting in Iran. So I really regret not to be able to be with you, because there are quite a number of talks that I would have loved to see. But I'm still somewhat happy that I'll be able to give my presentation, and I really hope to meet you physically in the near future. Okay, so what I want to talk about today is in the title, control of synaptic plasticity in deep cortical networks. So can you see the slides?
Yes.
Okay, great. So I'm going to start with a bit of a detour. Can you also see my mouse? I made it very large.
Yes.
Okay, great. So this is actually where I come from. I'm really interested in perceptual organization. So really how the brain understands complex pictures such as this one. For instance, if you see this picture, you probably don't have a hard time to see that this and this is part of the same zebra. And in the analysis of the visual scene, it doesn't really help that image understanding starts with fragmentation. So neurons at low levels of the visual system have very tiny receptive fields, right? That's not very helpful if you want to see that this and this is part of the same perceptual object. Now, what we have established in quite a number of studies is that what happens in the visual cortex when you see a scene like this, that those image elements that belong to the object that you're interested in are labeled by enhanced neural firing. And so basically, if this is the picture that comes in, and if you're interested in the central zebra, the visual brain creates a representation where all those image elements, all those neurons that code features of the zebra you're interested in have extra activity. Okay, now how is this related to plasticity? Well, the proposal that I'd like to defend today is that those features, those neurons that code features that are labeled, that they are eligible for plasticity, and those features that have not been labeled, that they are not eligible for plasticity. So that's basically the bottom line, what I want to kind of present today. Okay, so this is where we come from. This shows you the overview what I would like to talk about today. So I'll give you a very brief introduction on the role of feed-forward and feedback connections. Then I would like to say a few words about contour grouping. So actually that's where these labeling operations, that's where we discovered that this happens. And then I'm going to really go into plasticity. So I will talk about training neural networks. And in the last part of my talk, I would also like to say a few words about what we have learned of how this is really implemented in the brain. Okay? So let's start with feed-forward and feedback processing. So this is now a very old slide, more than 25 years old now, but it's really guiding a lot of my thinking about how this can all fit together from Velleman and Van Essen many years ago. So this is the Primate visual system. I think most of you know this slide, and also the next one. So this is if you take all the visual cortical areas, you can arrange them into this hierarchical scheme where information first enters in the primary visual cortex, then ascends to go to all these higher visual areas. And we know, of course, that low-level areas extract simple features and higher-level areas complex object categories. And so all these things happen fairly fast. Now, what is interesting is that this first feed-forward processing phase is followed by a later phase where feedback connections here in blue and lateral connections here in red come into play. And they allow for a sort of recirculation of the activity. And this is actually the phase where this labeling process I was just talking about takes place. Okay? Now, this is actually the process that psychologists call selective attention, and this labeling of image elements that they would call typically object-based attention. So to summarize then, based on this scheme, what you can expect from neurons at different areas. So if you have your electrodes in early visual cortex, then you find many neurons that respond to the onset of a stimulus. If you have your electrode in association cortex, you'll find these sensory neurons, but you'll find them intermingled with neurons that have memory activity. So basically they bridge the time between a brief sensory stimulus and a later response, even if the stimulus is no longer there. And then you also find these neurons that have what I call here ramping activity. So they might be coding for a particular feature and they have extra activity, they boost their activity if that feature becomes relevant to behavior. If you have then your electrode in frontal cortex, you have all of these as well. And then you find another category of cells that respond around the time of a movement. Okay, so for the purpose of my talk today, I'll kind of take all this intricate complexity and I'll map it onto this 3-layer feed-forward network. So where it's a sensory layer, association cortex, and this is motor cortex. And now we do know that we have these feedback connections from the motor cortex back to sensory areas. So as a result of that, you actually might expect to see some of the selection process also at the level of early visual cortex. And that's what I'm going to demonstrate. Even memory signals you might expect there, okay? Because you have these feedback connections. Okay, so this is basically the framework that I like to work in. So you can think of this as a sort of a primate Turing machine where input arrives and the input layer goes to the association cortex. Then you have the output layer, some say motor cortex. And it's convenient to think of all these output neurons as coding for distinct actions. So now one action will be chosen. So psychologists sometimes call this a production. And this may stand for an internal action. So it might feedback to update the activity here. Basically, it would be a shift of attention. It can also be an external action. So say an eye movement or a hand movement. So that will have influence, of course, on the activity pattern on the input layer. So now you have an update of the internal state. So you get the different selection signals, maybe even different inputs. And then on the next time step, the system might select another action, right? Updates into internal state, select another action, update its internal state, and so forth. So what is, of course, the challenge for us is to understand how the network can learn to select the right action and also to update the internal state in the most appropriate way. As a result of, for instance, during reinforcement learning, during feedback about the amount of reward that is gained. Okay, so that's the thing that I would like to discuss today. Okay, now to kind of illustrate this idea about feed-forward and feedback processing, I'll now make my short detour on contour grouping. And so I don't see all of you, so this is going to be a bit of a competition. So what I'm presenting here is 3 dots. One of them is red, one of them is green, one of them is blue. And in just a second, I'm going to present 2 curves. Now, one of these curves is going to make a connection between the red dots and either the green dot or the blue dot. So if you think that the red dot is connected to the green dot, you might want to raise your left hand. If you think it's connected to the blue dot, you might want to raise your right hand. Okay, and I can imagine you want to be just a little bit faster than your neighbor. Okay, are you ready for this? 1, 2, 3, go. Okay, I see some left hands. Yes, it's left. You go here and then you end up there. So this is the time to take revenge on your neighbor. So now you want to really be faster. So now we start from this side. Are you ready? 1, 2, 3. I see right hands. It takes a bit of time. Okay, so this is, I think, an example where you do a lot of recurrent processing for a very simple image. So what you're doing here actually is making a number of eye movements. So you typically direct your gaze to the red dot, and then you're going to make a sequence of eye movements over this curve, and eventually you will end up at the blue dot. Okay? But this is a stationary image, so you can only make saccadic eye movements. So what you'll do is move your eyes here, and then you make a saccade here. Now, how come you make a saccade on the right curve? Well, basically, before you make your saccade, you do a bit of mental tracing along this curve. Okay. Then you make your eye movement. Then you do a bit of mental tracing. Then you make your eye movement. Then you do a bit of mental tracing, and so on and so forth. And this mental tracing is actually the updating of the internal state of your Turing machine, and then you make an eye movement, which is the action. Right? So this is how it corresponds to what I was just talking about. Okay, how does this work then? Well, we did quite some work on this task in monkeys. So we asked the animals to look at the central dot in the center of the screen. Then this is a simpler version than you've just been doing. So there are two curves. One of these curves makes a connection between the fixation point and a larger red circle that's going to be the target for an eye movement. And this is a distractor. And then after a delay, the animal now makes an eye movement to the end of the target curve. And what we find in this task is kind of interesting. So we typically do multi-unit recordings from chronically implanted electrodes. So this is now the receptive field, and we are now recording in primary visual cortex. So first cortical stage of visual information processing. So here's a receptive field that's on the target curve. Basically, the animal is mentally tracing through the receptive field. Here, the same receptive field is on the distractor, because now the animal is mentally tracing this curve. And what we then see is the following. When you present the stimulus, so this is the first time that something appears in the receptive field. Okay, and then you get your feed-forward response. This is the information that is conveyed from the retina to the cell. Okay, this early phase does not discriminate between a target and a distractor. But then you wait a little bit, and then you see that activity elicited by the target is actually stronger than that elicited by a distractor. So this is recurrent process. Okay, and you get the same effect if the two curves intersect each other. So you get more activity if the receptive field's on the target. Okay, so basically what we see here in this task is that you can see, and I haven't shown you all the data we have on it. Basically what you see is that you get a propagation of the enhanced activity along this curve. So basically you see the gradual updating of the internal state of the Turing machine during this task. You can read it out from primary visual cortex. Now we have also recorded by now from higher cortical areas, not just primary visual cortex. So this V1, we recorded from V4, which is a high visual area, frontal eye fields, which is an area of frontal cortex involved in the generation of eye movements. The receptive fields are larger in V4 than they are in V1, and even larger in frontal cortex. And so here all receptive fields would be on the target curve. Here all the receptive fields would be on the distractor curve. Now this is a replication of what I've just shown you. So you get more activity in V1 after a delay. So this is the recurrent processing phase. In V4, you also get first the feed-forward processing phase that does not distinguish between target and distractor, and then the response modulation. And in frontal eye fields, you get the same. You get a first phase where the neural response is only determined by there's something appearing in the receptive field, and then you have to wait for a couple of tenths of milliseconds, typically 150 milliseconds or so. And then you see that activity elicited by the target curve becomes stronger than that elicited by the distractor. So we think actually that these activities then feed back to lower processing levels. And so the whole brain basically selects the representations that are being selected for an eye movement response. Okay? So we do see that basically these selection signals, they are presumably fed back from frontal cortex that makes the decision of what to do next to earlier processing levels. And this is the signal that we're going to use to give us a much better plasticity rule. That's the idea. Okay, so now I'm going to talk about plasticity. And I want to propose a role of these feedback connections I was just talking about in learning. And this work was actually hugely inspired by this book that I guess most of you will know, by Sutton and Bartow, published also about 20 years ago. And so now we're going to talk about this neural network, but now we're going to want to train it. So activity first gets propagated to an output layer. And as I did before, I'm going to assume now that units in the output layer, that they are coding for distinct actions. Now we're going to go to the reinforcement learning regime. So we're going to assume that the selection of actions is a stochastic process. So, of course, with those output units that have most activity or most input are most likely to win in this competition. So then, since the action selection is stochastic, maybe on some trial with this stimulus, this action will win, and the network will obtain a certain amount of rewards. On another trial with the same stimulus, the network may choose this action, receive no rewards, or choose this action and receive a lot of reward. And over its experience with this stimulus, the network will build up a certain amount of reward that is expected. And then we're going to compute the reward prediction error, right? People typically think it's dopamine, but it could also be another neuromodulator. So if now more reward has been obtained than was expected, we're going to release this neuromodulator as to increase the strength of the synapse between those neurons that were active, thereby increasing the probability that for the same stimulus, this kind of unexpectedly good action will be selected again. Okay, that's the whole idea, and it's fairly standard in the reinforcement literature. Okay. Now, if the network chooses an action that turns out to be disappointing, there will be another neuromodulator or another concentration of the same neuromodulator as to decrease the strength of these synapses. That's the idea. So this is fairly standard. This is a three-factor learning rule where synaptic changes depend on the activity of a presynaptic cell, on the postsynaptic cell, and on the level of the neuromodulator. And that typically works fairly well for the second layer of connections here. So here, you're going to increase the strength of this synapse. And in this situation, you want to decrease the strength of these synapses. Here is only pre- and postsynaptic activity. So this is the only synapse that will decrease in strength. Okay? Now you also might want to decrease the strength of these synapses, right? And the problem here is that if you're going to do that, you might also want to decrease the strength of these synapses because the pre- and postsynaptic activity here is the same as it is here. And the level of the neuromodulator here is also the same as it is here, right? But these synapses have no responsibility for the choice of this action. Now the same here, you might want to increase the strength of these synapses, but again, then you would also have to increase the strength of these synapses, right? So the conclusion is something is missing from this scheme, and this is typically known as the credit assignment problem. And the proposal is that this is where you can use the feedback connections. And the idea is fairly simple. If an action gets selected, we know there are feedback connections. We've just seen that they increase the activity of neurons in visual cortex. And the idea is to use this as an eligibility signal. So only those synapses that get the feedback, they are the ones that are allowed to change in strength. Here they would decrease in strength. And in the next trial where this action is selected that is better than expected, then those synapses are the ones that are going to increase in strength. Okay? So now you have a 4-factor learning rule. So we added the feedback from the response selection stage as a 4th factor. And actually, we have some data from the lab where we demonstrate that feedback connections seem to rely to some extent on the activity of NMDA receptors, which are known to get plasticity. And I will say a bit more about that in a little bit later. Okay. Now, if you then do the math, then you'll find that if you do it right, you can actually get the same synaptic modifications as are prescribed by error backpropagation if you do stochastic sampling across the different action possibilities. Okay, so this is a learning rule that is biologically plausible, we believe, and that gives you the same synaptic modifications as error backpropagation. Okay, so this is the whole idea. We are going to use these feedback connections to gate plasticity. Okay, now let's give you some intuition of how this might then work. So I'm going to model this task that was studied by Friedman and Assad a couple of years ago. And in their task, they had monkeys looking at a fixation point. They saw a cloud of moving dots. They were moving in one of 12 directions. And they were actually separated into 2 categories. So the motion directions in red, indicated in red here, there were 1 category. And those indicated in blue here, they were the opposite category. So then the animal was still maintaining fixation, and he had to remember the category of the motion that he just saw. And then he saw a second cloud of moving dots, and he had to judge whether it was the same category or whether it was the other category. If it was the same category, he had to release a lever. If it was a different category, he had to hold a lever. And what is nice about its design is that you can have motion directions that are from the same category, but that are very different. And that you can have motion directions that are fairly similar, but that belong to different categories, effectively dissociating motion similarity from category membership. Okay? So, and what they then found, recording an area LIP, I would call it association cortex, is that many neurons there become tuned to category membership. So this is one example neuron that really liked the blue motion category. So this is a sample phase. You see that whenever there was a sample from the blue motion category, there was a pretty good response. Now, the moment you cross the category boundary and you go to the red motion, you see that the response will drop. It will stay low. And the moment you cross the other category boundary, the response will increase again. Okay, so basically this neuron was most sensitive to changes in motion direction that straddles the category boundary. And this is what they found across quite a number of LIP neurons, that most of these cells were most sensitive to changes in motion directions across the category boundary. So in other words, these cells became tuned to categories. Okay, so how does the learning scheme that I just proposed produce such a tuning? Well, I'm first going to simplify the tasks substantially. So to give you an intuition of how this might work, I'm going to have a very simple task where there's simply a cloud of moving dots. And the only thing that our model has to say whether it's category 1 by making one response or whether it's category 2 whether it's the other response. So now there's not a necessity to keep the previous category in working memory and then make the comparison. I will come back to that later. Okay, this is the simple version. Okay, now here is our neural network. In the input layer, neurons that are tuned to motion direction. So we're going to present a stimulus. Then we get the pattern of activity on the input layer, of course defined by the tuning of those cells. We start with completely random connectivity, so you get some pattern of activity on the association layer. And here with the output layer, and since action selection is stochastic, sometimes the network will choose category 1. Now let's assume that this is the right action. So then there will be the feedback from the response selection stage, making some of these feed-forward connections eligible for plasticity, sensitive to the neuromodulator. And then the neuromodulator is released. It is better than expected, so these synapses that have been tagged will be increasing in strength. Okay? Now on another trial with the very same stimulus, the network may make an error. It will choose category 2. Other synapses will be made eligible for plasticity. But now you have the other neuromodulator or the other concentration of the same neuromodulator as to decrease the strength of these synapses. Okay, now what is nice is that you'll see that such a simple network will learn such a fairly simple task in a couple of maybe 100 trials or so. And then if you look at what these units become tuned to that are in the association layer, you'll see that they also become sensitive to the category boundary. Okay, so this will give you some intuition of how this will work might work in the cortex. Okay, so such a fairly simple learning rule, we call it AGREL. We will see that neurons become tuned to those features that are important in the stimulus response mapping, and neurons will code for the boundaries between categories. Okay, so that was quite some time ago, and now we wanted to generalize this scheme to more complex tasks in which you sometimes have to do quite a number of actions before you get your reward. And we chose this delayed saccade, anti-saccade task because it also has this exclusive R structure and also the requirement to keep something in working memory. So this is how it works. The monkey, this was typically tested in monkeys, looks at the fixation point, then he sees a cue. The cue is either on the left or it's on the right. Then there's a delay. So now the monkey has to remember the location of the cue. And then as a go signal, the fixation point switches off, and now the animal has to make an eye movement to the remembered location of the cue on pro-saccade trials with the green fixation point. but it actually has to go to the opposite direction if it's an anti-saccade trial with a red fixation point. Okay? So we want to train our neural network to solve this task, and now we are not going to give it reward immediately. So now we have to go to a form of reinforcement learning where it's possible to delay the reward. And we're going to go to a form of reinforcement learning that's known as Q learning. I think most of you will be familiar with it. For those of you who are not, I'm not sure whether they are in the room. I'll quickly guide you through this. So this is just a task in which you have to take a number of actions before you can get your reward. So and this is a very simplified task in which you always start in state 1. You can choose one of 3 actions. Then if you choose action 1, you will go to state 2. You can choose 2 actions. Now suppose you choose action 2. This is actually the only action that eventually will give you a reward. And of course, the challenge is to learn to take action 1 in state 1 because you're never going to be confronted with a reward immediately after that action. So this is Q learning. It was proposed many years ago by Watkins and Dayan. And the trick is that you're going to remember, if you go from state 2 to action 2, you happen to stumble on the reward, then you're going to remember that if you take action 2 in state 2, that has a positive reward prediction. Okay, so and you store a positive value for this Q. Now, if in a later trial you are in state 1 and you happen to choose action 1 and you then go to a state 2 where you now know from here there's a positive Q, we're also going to increase the Q of this state 1 action 1 combination. So basically the network is now backtracking those state action combinations that eventually will lead to reward. Okay, that's the only thing that the network has to learn. Okay, and it turns out that our model is completely consistent with the learning of action values, or our Q values, I should say. Okay, and this model is called Augment. So now we're going to train it on this delayed saccade anti-saccade task. So this is the network. Here's the input layer. There are 3 units that you're already familiar with. They're just kind of hidden units. But we now also introduce a form of a unit that can memorize activity. So if there's a brief stimulus, the unit goes up in activity and it stays active even if the input is no longer there. It's sort of an LSTM unit, but it doesn't have any gates. So it's a very simple LSTM unit. Right? So we simply assume that they exist in the brain. Of course, we know that there's persistent activity. Okay? And actually, in the simulations, there will be 4 of those units with the memory property. Okay? So this is the actual simulation. So there will be a unit that computes the Q value for looking straight ahead. So that's F for fixation. There's a unit for the Q value for looking to the left. Here the network is actually looking to the left. And there will be a Q value for looking to the right. This is the action that is actually selected by the model. So typically, of course, it chooses the action with the highest Q value, but it also needs to explore a little bit. Then in the input layer, we have a unit that codes for the green fixation point, one that codes for the red fixation point. So there will be an anti-saccade trial required, and a Q on the left and a Q on the right. Now we also have an on version of these input units, and an on unit is when the fixation point switches on. The on unit is only on for 1 time step and then switches off again, although the fixation point is still visible. So we know that these units exist in the brain. There are also off units. So an off unit would, for instance, respond if the green fixation point appears. It doesn't respond, but it only responds the moment it switches off. And we also know that these off units exist in the brain. Okay? So now we start with completely random connectivity. This is the number of trials that have elapsed. And let's see whether this works. So basically, this is what is visible on the screen. So this is the green fixation point. The model should actually start by attaining fixation, but it does not. So this is very familiar for those of you who have trained monkeys. So this is when you bring your monkey the first day to the lab. The animal has no clue, right? It's just looking around. Okay? So then after a little bit of experience in this task, this is after 3,000 trials and a few, you'll see that the network now really starts to develop. And the blue connections are excitatory and red connections are inhibitory here. And so this is what happens. So you see that the network, when the fixation point appears, typically attains fixation. And So this would be an anti-saccade trial. Model makes an anti-saccade. This is a pro-saccade trial. Model makes a pro-saccade. This would be an anti-saccade trial. And an anti-saccade. So it looks good, but actually the model is still making quite a number of errors. This would be an anti-saccade trial, but it makes a pro-saccade. This is a pro-saccade trial, and it makes an anti-saccade. Okay, so you see the network gets the structure, the time structure of the test, but it still makes quite a number of errors. Now, about 2,000 trials later, you'll see the network is now perfect. It's always getting it right. So this is a pro-saccade trial, makes a pro-saccade, anti-saccade trial, makes an anti-saccade. Okay, well, this goes on and on. And so basically you see now that what has happened here in this network, somehow it incorporated all the information that is necessary to solve this task. And this network has really only 7 association units. We could probably even decrease the number a little bit. So the network learned to wait for the fixation point, direct gaze to the fixation point, wait for the cue, remember the location of the cue, and then if the fixation point switches off, depending on the color of the fixation point, make a pro-saccade or an anti-saccade. So I think this is quite remarkable that such a simple network with a biologically realistic learning rule can do all these things. Okay? Now then, of course, it's interesting to look what these units become tuned to. And this is just one example of those units that have a memory property. So you see that this unit switches on with the cue, so the spatial cue, it stays active during the delay because it has this ability to maintain activity, although there's no input anymore. And then it actually has an extra boost in activity when the fixation point disappears. And you see that this cell is much less active on the pro-saccade trial, so it has what some people have called this mixed selectivity that is required to solve such an exclusive OR task. Okay? So it can solve this saccade and saccade task, but what is interesting, we can take the very same network and basically the very same learning rules, same learning parameters, and it will also learn the full version of this delayed match to category task. So it can also remember the category of the stimulus and then also decides whether a second stimulus is matching or non-matching and make the appropriate response. It can do fairly complex decision-making tasks. So this is a task in which symbols were presented successively and they gave some probabilistic evidence whether it would be a good idea to make an eye movement to a green circle or to a red circle. This is another fairly complex task in which there are 2 moving sets of dots, and then there was a sort of a change in motion in either location, but only the location that had a particular color mattered to the monkey. And these colors were in a rank order. So suppose the rank of red was higher than the rank of blue, then the model should only respond to motion changes in the red, but there might have been another color, say green, that if paired with blue actually would have been, then blue would have been relevant, and then the model should ignore the green, but attend the blue, and so on and so forth, right? And the model learns that. Now with some additions, and now we really need to include recurrent connections, so also feedback connections that do not only gate plasticity, but that also kind of propagate activity. And here we kind of, in this paper, we demonstrated that this is possible. Basically, it gives you an implementation of this so-called Pineda-Almeida algorithm. We can also train a model using a biologically plausible learning rule to trace a curve or to do other tasks that require perceptual organization. Okay, so this is a fairly general framework that allows neural networks to do many, many different things. Okay, so now in the last, let me see, maybe 10 minutes or so, I would like to say a few words about the neurobiology of all of this. So how plausible is this then? Right, so what we need here is, in addition to the pre and post synaptic activity, we need the gating by neuromodulators, and we need the gating of plasticity by feedback connections. Now, the effect of neuromodulators, I think, is fairly well established. And right now, I would like to focus more on this idea that feedback connections gate the plasticity. And I know in the workshop there are other people who also have proposed that. So maybe we might even agree on this. Okay, so but that's what I want to show some evidence in favor of. And I want to review data that were from the group of Anthony Holtmaat. The first author was Gambino in a paper published about 4 years ago. where they recorded from layer 2-3 cells in the barrel cortex of rodents. And what they did here is what they call repetitive whisker stimulation. So they kind of move this whisker along for some time. And this basically shows how the information goes from the thalamus then to a neuron in barrel cortex. It, of course, enters in layer 4, and they record it from this cell in layer 2-3. And basically what they find is that this repetitive whisker stimulation increases the strength of the synapse between layer 4 and layer 2-3, which is what you see here. So this is the postsynaptic potential, and after a little bit of this repetitive whisker stimulation, you see that the amplitude increases, which is also what you see here. Okay, now since they have an electrode in the cell, they can also look. Oh, this is basically quantifying the degree of LTP. So it starts at 100%, so you get about 20% extra strength of the PSPs. Now here they record it intracellularly, and what they then see is when they kind of give the whisker stimulus, you get this first early PSP, and then you get this very prolonged NMDA-related plateau potential, and they basically find that in the apical dendrites of these cells, there's a lot of calcium influx. Now, how do they demonstrate that this is dependent on NMDA receptors? Well, they can give an NMDA blocker, the AP5, and basically what you then see is that this early PSP remains, but this later plateau potential disappears. Okay. Now, they then demonstrated actually that this late plateau potential, oh yeah, I forgot to say, so if they then block the NMDA receptors, maybe not so surprising, they also take out the LTP. So now there's no plasticity anymore. I guess that is what many people would have expected. Somewhat unexpected is that they basically find the source of these plateau potentials in a higher order thalamic nucleus known as POM. So POM is a higher order thalamic nucleus, unlike VPM, which is a primary nucleus, and it has a pattern of connections back to S1 that has this feedback-like topology. It goes, for instance, to layer 1. Now, if they now block activity with muscimol in POM, they block the cells here, and you see that now the plateau potentials are much less pronounced, and this manipulation actually also blocks the LTP. Okay, so this, I think, is direct proof that feedback connections can gate plasticity of feed-forward connections. Okay, so this is basically the second point I wanted to make. So now let me then summarize everything and let me put it all together. So here we have our neural network activity. Let's focus on these 3 neurons here in the association cortex, right? So they are magnified here. So activity first gets propagated from the input layer and it passes through the association layer. Okay, that's what you see here. So the units start to evoke or produce spikes. And this is the early phase of the PSP, and this is actually the feed-forward response in visual cortex I was talking about. Then a little bit later, one of these actions gets selected, and it starts to produce feedback back to these neurons. Okay. And we now know that this is associated with calcium events in the apical dendrites. Okay, so that's what you see here, calcium events in the apical dendrites. And now we're kind of looking at the late response of these PSPs, and we're looking at this phase where the neural activity, spiking activity, becomes modulated by attention. Okay? Now something needs to happen in this cell to make the feed-forward connections eligible for plasticity. And our assumption is that what is produced here is some biochemical marker that we call synaptic tag, or people have called this synaptic tag, or an eligibility trace. Right, so these calcium events become replaced by these tags. Okay, so now the activity is the network is maybe doing something else in the meantime, and maybe producing another response that's going to produce a reward prediction error. And this reward prediction error now needs to interact with these synaptic tags. And if it's better than expected, it needs to strengthen the strength of these synapses. Okay, so now this synapse becomes stronger. Okay, so this is what you need. Okay, so to say this another way, we need 2 events. The first event is the production of this synaptic tag or eligibility trace that depends on the calcium events in the apical dendrites. And there's actually some nice evidence of how this might work. So this is from a paper from Yagishita in the group of Kasai, published about 4 years ago, where they kind of proposed that what you need here in this phase of the process, you need activation of NMDA receptors, calcium influx into dendrites, right? So this is now very, very familiar to us. And then a little bit later, you need the reward prediction error. And in this paper, actually, they demonstrate that there can even be a little bit of time between the moment of this production of this eligibility trace and the delivery of dopamine. Okay, and that then activates a different pathway. So it starts with dopamine. It goes through several intracellular messengers, cyclic AMP, phosphate kinase A, and so on and so forth. And these two pathways are proposed to converge on a protein known as CAMcase2 that is known to phosphorylate itself. And together with some protein synthesis, this is then proposed to give rise to an increase in synaptic strength. Okay, so this is how this all might fit together. Okay, so this is what I wanted to say about the neurobiology of synaptic plasticity. So I think that this puts us in a very interesting place because we now really start to understand at the molecular level, at the cellular level, at the network level, and also at the computational level, how all these pieces of the puzzle fit together, allowing the brain that has, of course, many layers between input and output, to give meaningful changes in the strength of synapses that allows the Turing machine to train itself. Okay, so a few conclusions. In the beginning, I kind of posited that contour grouping is associated with labeling of contour elements with enhanced neural activity. And the idea is that this labeling signal that we know as selective attention enables plasticity, making some connections sensitive to the reward prediction error. We see that how the conjoint influence of attention and reward on plasticity allows the implementation of error backpropagation in a biologically plausible manner. And I would like to conclude by saying that these considerations make deep learning even more relevant for our understanding of learning of the brain. And these are the people who contributed to the work. Pieter Lam and Henk Spekerijs were involved in the early contour grouping work together with Paul Kayat, Arzu Porsmali, and Jasper Poort, who made the recordings from the higher visual areas. The theoretical works was started off with Arjen van Ooyen and together with Lawrence Watlin. And the augment model for Q-learning was done by Jalbert Rombouts together with Sander Botté. And the model in which we trained recurrent networks with a biologically plausible algorithm was done by Florian Raudis and Heiko Neumann. And the work on how this all fits together with what we know about the brain was done together with Anthony Holtman. Thank you very much.
And perhaps for one or two questions while Christos is setting up the next talk. Oh, we can't set up because of the remote connection. Are there any questions?
Hi, Pieter. Very nice talk. I have one question on this data of Yagishita that you mentioned there for particular type of synaptic connection from cortex to striatum. So does this fit into the network model, or can you kind of reconfigure your network model that you have this type of gated plasticity only for this type of synapses?
Yeah, you're mentioning that that this happens in the striatum, right?
Yeah.
I think that's a really important finding. I would actually suspect that similar forms of plasticity might also be happening at cortical synapses, but I don't know, right? So that's I think we start now to have some insights in how this all might fit together, but there are also many open questions. And I would like to speculate that these loops through the striatum and then back to cortex might also play a role in producing persistent activity. But of course, that's entirely speculative. There is definitely some evidence from the Svoboda lab that something similar is happening between thalamus and cortex, right? That produces persistent activity.
Can Pieter hear me?
Yes.
Okay. Pieter, this is Bartlett Mel. So there's a little bit of a tension because we've seen presentations about reinforcement learning on the machine learning side, and the goal is to do these extremely difficult, solve these very, very difficult problems that might require very, very deep networks. And we heard Timothy yesterday or the day before talk about how the type of algorithm that you're mentioning works well for small networks and works poorly for big ones. And so I'm kind of just wondering if a monkey, well, you know, you have a much better sense of what a monkey is capable of doing through reinforcement learning. And one question that I have is, do you think that this type of, well, relatively shallow learning thing, you know, you have one layer of, from what I can see, it looks like you have one layer of weights that are between the inputs and the outputs that are kind of hidden and have to be supervised by this reinforcement. Do you think that that can explain essentially what a monkey can actually do, as opposed to, say, play Go or some other very, very complicated tasks? Is this going to be enough power? Is a small network enough to explain monkey reinforcement learning?
So we can generalize this learning rule to deeper networks. So in principle, the answer is yes. And now the question is, how does the network compare to learning in monkeys? The network is about, well, it's difficult to estimate, but typically it's about 10 times as fast as the monkey. So monkeys learn much slower than the networks, but who knows, you know, maybe these guys are thinking about, they're distracted, and of course the displays that they see are much more complex than these highly abstracted type of input that we provided to the network. So, but I guess the good news is that the learning rule that we have is not much slower than learning in monkeys. I'm not sure whether what Finnick talked about. Wolfgang Maas gave me a link to a nice paper that is on arXiv, where they kind of demonstrate that humans can, or actually there's a way of getting reinforcement learning to also learn faster than is dictated by the more slow synaptic changes. And I think our framework is quite compatible with that idea. So if, for instance, if there are two contexts, say there's context A where stimulus 1 requires response 1 and context B where stimulus 1 requires response 2, then by kind of monitoring and representing the error that you got on the previous trial and also memorizing what you just did on the previous trial, you can actually quickly switch context also in a network like we have. So in those situations, a monkey would also actually learn that there are two contexts, and that based on his previous experience on the last few trials, he probably also learn which context he is in. And in those situations, I could imagine that our learning rule would also produce a similar result.
All right, so we're a few minutes over time, so I think we'll have to stop questions now and thank the speaker.
I really hope to see all of you in the near future physically. So I'm applying for visa now. So, okay, bye-bye.
Our next speaker is Christos